Hey, it's Mr. Shrum, and we are going to talk about cell division today and meiosis, mitosis and meiosis. There are two pathways to cell division. This is a process in which cells divide to make two or more cells. We have mitosis on the left and meiosis on the right. Mitosis is when a cell divides and creates two cells with the exact genetic information of the mother cell. They're like identical copies. Therefore, the new cells are capable of dividing again. So we have replication, cell division. We have 2N daughter cells. And that's just how we refer to cells that have been divided. Meiosis is when a cell divides and creates two cells with only half the genetic information of the mother cell. Therefore, the new cells must be fertilized before they are capable of dividing again. And there are 7 phases to cell division. We have interphase, prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. Interphase is when the cell prepares for dividing by growing larger and making all the necessary materials. More organelles are produced, DNA, deoxyribonucleic acid, in other words, which contains the genetic makeup of the cell and blueprint for creating new cells, makes 2 exact copies of itself. Prophase is the first phase of mitosis. During prophase, the nuclear membrane, the membrane that encloses the nucleus, begins to disintegrate. Centrioles begin forming spindle fibers and move away from each other. Prometaphase is the phase in mitosis characterized by chromatin, which houses the genetic material such as DNA, compresses into shorter and thicker rods called chromosomes. The 2 pairs of centrioles move to the 2 polar ends of the cell. So you see how they're separating towards the poles. And then you see the chromosomes forming. Metaphase, this is a misprint, but that is the phase in mitosis when the chromosomes are arranged along the middle of the cell. So you see how everything's aligning right down the middle. Hint, metaphase, think of middle phase. The centrioles are now in place at the polar ends of the cell. Spindle fibers attached to the centromeres of the chromosomes. Anaphase. Anaphase is the phase in mitosis when the chromosomes are pulled apart. Think of anaphase and apart, apart. The spindle fibers contract or shorten, pulling the chromosomes apart. See how everything's getting divided, pulled apart. Telophase is the final phase in mitosis. Telophase, think of tail end. So T, tail end, telophase of mitosis. And a cleavage furrow is formed as the cell membrane pinches in. A new nucleus is created in each of the new cells. And then finally, cytokinesis. That's the final phase of cell division. And it's when everything else in the cell is divided, resulting in the completion of 2 new identical cells. So these were the 7 phases of cell division for mitosis. And then we're going to take a look at meiosis as well. Meiosis is a little more complicated. Mitosis, like we just looked at, that's when a cell divides and creates two of the exact same cell. They are exact copies of the mother cell. Therefore, the new cells can divide again on their own. Meiosis is different, and it creates two cells with only half the genetic information of the mother cell. These cells need to be fertilized before they can divide again. Meiosis begins very similarly to mitosis, but it goes through each phase twice. The phases of meiosis include interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Meiosis 2, which the cell repeats prophase through cytokinesis again. So it goes through it once and then again. You can kind of see that in this diagram. Interphase. That's when the cell prepares for dividing by growing larger and making all the necessary materials. Like we described before, more organelles are produced. DNA contains the genetic makeup and blueprint for creating new cells, and it makes two exact copies of itself. So DNA makes two exact copies of itself. Prophase is the first phase of meiosis. During prophase, the nuclear membrane, the membrane that encloses the nucleus, begins to disintegrate. Centrioles begin forming spindle fibers and move away from each other. Chromatin, which houses genetic material such as DNA, compresses into shorter and thicker rods called chromosomes. Metaphase. Metaphase is when the chromosomes are arranged along the middle of the cell. The centrioles are now in place at the polar ends of the cell. Spindle fibers attach to the centromeres of the chromosomes. During metaphase 1, chromosomes are composed of 4 sister chromatids, known as a tetrad. Overlapping pieces can swap chromosomes, and this is known as crossing over. Anaphase is when the chromosomes are pulled apart, A for apart, and the spindle fibers contract, shorten, and then this action pulls the chromosomes apart towards the poles. Telophase, this is the tail end of the division part, and that's when a cleavage furrow, we call that the cleavage furrow, is formed as the cell membrane pinches in, and then a new nucleus is created in each of the new cells. Cytokinesis, that's when everything else in the cell is divided, resulting in the completion of 2 new cells. At the end of meiosis 1, this first cell division, there are 2 diploid cells. This means that each new cell has the complete 46 chromosomes. 46 chromosomes come in pairs, so we have 23 pairs of chromosomes that humans have. Therefore, there are 2 copies of their DNA. Copies. Think of meiosis as forming 2 of everything. Meiosis 2. Meiosis 2 begins, and both of these 2 new cells will go through the following phases again: prophase, metaphase, anaphase, telophase, cytokinesis. At the end of meiosis 2, the second cell division, there are 4 haploid cells. So meiosis 1, at the end, you have 2 diploid cells. At the end of meiosis 2, you have 4 haploid cells. Haploid cells means that each new cell has only half of the chromosomes that humans have, which is 23 chromosomes. Therefore, there is only one copy of their DNA. These new haploid cells in humans are known as gametes. Meiosis for human reproduction. Humans use meiosis in order for reproduction to occur. Meiosis occurs in females during the production of egg cells, ova, during oogenesis. Each egg has one X chromosome and 22 other chromosomes. Meiosis occurs in males during the production of sperm, cells during spermatogenesis. Each sperm has either one X or one Y chromosome and 22 other chromosomes. When fertilization occurs during reproduction, the sperm's DNA is transferred to the ovum. This creates a zygote. And this zygote has a full set of 46 chromosomes, which is 23 pairs of chromosomes. The zygote then grows and develops into an embryo by the process of mitosis. So now we're back to mitosis. Genetics. Genetics is the study of how characteristics and traits are passed from parent to child through our DNA. When a zygote is formed, it is composed of two gametes fused together. This results in the zygote possessing two of the same gene. One of these genes may be dominant, meaning it is the more influential gene. It dominates compared to the other gene. One of these genes may be recessive, meaning it is not as influential. We can use genetic diagrams to trace how a specific trait or characteristic is passed down from parent to child. Visit the field trip to see examples of how this is accomplished. Most characteristics occur as a result of a specific gene or a combination of genes. However, determining the sex of a child is controlled by a whole chromosome. Individuals with two X chromosomes, XX, are female. Individuals with one X, one Y chromosomes are designated as male. Now, this diagram demonstrates the possible ways the X and Y chromosomes can combine. It also explains how each time you have a baby, you have an equal chance of having a boy or a girl. Meiosis occurs in humans to allow reproduction to take place. It results in 4 haploid cells or gametes. When fertilization takes place, the two gametes combine, creating a zygote with all 23 pairs of chromosomes. The zygote will continue to grow and develop using mitosis to develop into an embryo. And that's cell division. For mitosis and meiosis, I would go back through and make sure you understand what is happening. Mitosis is creating copies that are exact copies of the mother cell, whereas meiosis, you're creating cells that have half the genetic information, and they will need to fertilize with another cell to create the zygote that can grow and develop into an embryo for human reproduction. But that's the end of this lesson, guys. I hope you learned something. And if you have any questions, don't be afraid to ask me in Canvas, and I will see you all next time. Bye-bye.